Welcome to week number 4, everyone. This week is our week on metabolism and also some photosynthesis, especially if you're in the lecture section. You'll talk some more about some photosynthesis, but for us this week we're going to concentrate on metabolism in lab. So let's see what we've got going on. Yeah, I can't get it to go to the next slide. I'll get it. Thank you. So, last week, what did we do? We learned about diffusion, how that iodine solution diffused into the potatoes, osmosis. You did the experiment with the tubing, the dialysis tubing that simulates cell membranes. You learned about cells. You learned about viruses. You made your own. Of course, you didn't make them, but anyway, you designed cells and viruses so that you get a feel for what's inside each of these different types, the prokaryotic, the bacterial cells, and eukaryotic cells like ours, and then what's in a virus. Okay, so this week is all about cellular respiration and metabolism. So first of all, metabolism is all the chemical reactions that take place in organisms/cells. Okay, that's metabolism. Normally, you think of metabolism as your body speeding up its chemical reaction so that you burn energy faster and you lose weight, that kind of thing. But that's not really what metabolism means. It means all of the chemical reactions taking place in your cells. Well, some of those chemical reactions are where you break down food to produce the subunits that you need to make those macromolecules, but also energy in the form of ATP, okay? So if you are aerobic, you do aerobic respiration. We are aerobic. Lots of organisms on the planet are aerobic, including plants. So we do this process called cellular respiration, and in particular, aerobic respiration, where we use oxygen to break down our food molecules to make lots and lots and lots of ATP molecules. And ATP is stored energy that then is released to let enzymes do their work. Okay? We'll also mention photosynthesis, and that has to do with our peas, and I'll explain that in a second, what that's got to do with your peas. So photosynthesis is the process of making glucose from carbon dioxide, water, and sunlight energy. So you use sunlight energy in order to make the glucose. And in lecture, which I'm going to mention in my video for that, if you're in my lecture section, there's an assignment that you have comparing cellular respiration and photosynthesis. Are they opposite reactions? Are they complementary reactions? So let me make the point right here. Plants that do photosynthesis also do cellular respiration. Wouldn't this make sense? So what's the point of photosynthesis? To make glucose, to make food. So plants make their own food. There are plenty of other organisms on the planet that do photosynthesis, but just use plants as an example. So plants use sunlight energy to make glucose, which is a sugar. That's their food. All right, what's the point of making glucose if you don't ever break it down? Right? So plants have to do cellular respiration. They make ATP from glucose that they also make. Okay? They make their own food and then they break it down to gather up as much ATP as they can from each glucose molecule to do all the chemistry that they want to do. So plants do both, just as a tip. Okay, let's talk about the lab for this week. The lab this week is going to take several days, and I mentioned last week that you ought to start this experiment by Thursday. Don't expect to do this Saturday night or Sunday night, because that is not going to happen. You got to get the peas soaking for several days before you start the experiment. So here are my tips for this lab. You're going to soak the peas overnight in non-chlorinated water. Make sure you use bottled water of some type, filtered water, you can even use tap water. if you will get a big old container of it, pot of it, whatever, set it out for a couple of days, all that chlorine will evaporate. Okay, so you got lots of choices of water, but do not use chlorinated water. Soak the peas overnight per the instructions. Soak them in water. Take them out the next day. Put them on wet paper towels. Also, do not use chlorinated water for the paper towels. You put them in damp paper towels, in between 2 sheets of damp paper towels. Put them in a baggie or something like that, and put them in the dark. Now, here's where the photosynthesis is going to get mentioned again. You want to put them in the dark so that they don't do photosynthesis. They shouldn't be starting to do that yet. You just want them to start metabolizing, starting their biochemistry, starting their cells going again, because realize a dried pea is a dried plant embryo. So we need to rehydrate it to make it come alive and make it start doing metabolism again. But we don't want it to do photosynthesis. All right, the next day, so now we're on day 3, you can set up the respirometers. And that's the little gadgets that you're going to set up. You're going to follow the instructions and make these little respirometers to see how much oxygen the different peas, the germinated and ungerminated peas, take in. So if they do metabolism of any type, if they do cellular respiration, they will consume oxygen. They will pull it in, right? And so you should see the dye on the end of the little pipette moving in toward those peas if they are taking in oxygen and doing some metabolism. So that's what you're looking for, and that's what you're measuring. So that is day 3. You have to work on the peas for 2 solid days before you can even set up the experiment. All right, you are going to use a chemical called potassium hydroxide. It is very caustic. That means it will burn your skin. So be safe, please. Follow the instructions. Follow them carefully. Wear your gloves, wear your goggles. Don't have children or pets around because it is caustic. All right, and then after you finish your experiment, you do this timing for a certain period of time. I think it's 20 minutes in 5-minute increments. If you see that your liquid is moving really fast, then I would start recording every 1 minute. Okay, when I did it, I saw mine moving that fast. And by the time I got to 10 minutes, it was done. So if you see that, just change the time intervals. That's perfectly fine. Okay, adjust. Use your lab report forms. Make sure you do that and combine everything that's required into that one report. I shouldn't have to go to 3 different places to see your pictures and a table and your explanation and all that. And also realize the last question in that lab report is you explaining the biology behind what has happened. I don't know if that's really clear in the question that's asked, but that is what I'm looking for, because isn't that the whole point of this experiment? For you to do something, perform something, and then to learn something about metabolism, cellular respiration. Yeah, that's the whole point of it. So make sure on that last question that you're explaining the biology behind what has happened. All right, I have also posted in this announcement how to use parafilm, because it tells you to use it during this experiment, but you have never seen parafilm before and you don't know how to use it. It is a miracle material. I wish that they would sell it for household use, because I would buy it by the box because I love it. Anyway, I made you a little video on how to use it, and I am putting that in the announcement. And as always, let me know. Here I am in the branches of the tree, which I'm okay with. Anyway, let me know if you need any help during this week. Email me, put a message in the general discussion board post. Whatever you need, just let me know. I'm here for you. I want to help you. So let me know if you need something. And with that, I want you to have a wonderful week.